All right, Science 30. So we are now going to discuss cell division specifically, so mitosis as well as meiosis. So mitosis is where we're going to go into the most detail here in Science 30. We've got four phases preceded by what's called an interphase or a before phase. So interphase is like this preparatory phase. Things are copying, things are growing, things are synthesizing. We're starting to double up all the information because remember the end goal of mitosis is to copy everything inside that cell, all the information in those chromosomes, and then divide it so you have two identical brand new cells. So we're multiplying, we're copying. And so then you're going to enter another four phases, and they're as follows. So this is, I like to call it PMAT. So prophase, metaphase, anaphase, and telophase. And we're going to go through each of these very specifically. But I really like this diagram because it's really your one-stop shop here. So prophase is the chromatin, which is just chromosomes more tightly bound up, and they're going to sort of um, condense more. So you get those classic-looking X-shaped chromosomes. When a cell is normally hanging out, chromosomes don't really look like X's. They're more like spaghetti. And when the cell wants to divide, then it sort of tightly winds up the chromosomes to look a little more like that classic X, so that information is easier to move from each part of the nucleus. And so during metaphase, everything lines up along the middle. So M, metaphase, M for middle, the middle of the cell. During anaphase, everything tears apart. So A for apart, because everything's going to tear apart. So half the information is going to go to one side of the cell, the other half of the information is going to go to the other side of the cell. And then in telophase, the two cells start to tear or rip apart, and you'll ultimately end with two separate identical cells. Okay, here's another diagram of the two. Feel free to pause this video at the end and take a picture of screenshot of this diagram. So we'll start with interphase. Interphase is our preparatory sort of phase, and so we've got these things. We've got the nucleus, chromatin, which is loose genetic information. We've got something called the centriole, which are little things that have projections that come out that grab the nucleus. And this is just preparing for interphase, preparing for meiosis. The next step is prophase. During prophase, you're going to have spindle fibers form. So these little centrioles are going to start releasing these fibers that are going to attach to the individual chromosomes, which are going to be visible in a second. The nucleus or the envelope or the layer around the nucleus is going to disappear so that the chromosomes are accessible. And we start seeing the classic X shape of chromosomes. The next step is metaphase. Metaphase for middle. So during metaphase, and I moved it over here so you can see it better. During metaphase, all those tightly bound chromosomes have now lined up in the middle of the cell. Those centrioles have released the spindle fibers, and notice that there's a spindle fiber attached to every single half of each chromosome. And in the next phase, anaphase, we are going to pull apart. Here it says action, but I prefer the word apart. Anaphase, you're going to pull apart. So look at that. Those spindle fibers are yoinking on each individual chromosome and they're pulling them apart from the middle. And so you have a separation of each chromosome. Ultimately, half the information is going to end up on one side of the cell. The other half of the information is going to end up on the other side of the cell. The nucleus starts to reform around each side, and the actual cells start to split or tear, T for tear apart. And you'll have, at the end, two brand new identical cells. Boom. Mitosis is done. You have now gone from one, in a human, 46 chromosome somatic cell and created two identical 46 chromosomes somatic cells. So the process is interphase, prophase, metaphase, anaphase, telophase, or in other words, IPMAT. Okay, here's a little practice activity. See if you can identify what the different stages are on the left-hand side and on the right-hand side. The right-hand side might be a little harder because this is real images, and these are drawings on the left. Okay, here's the answers. So this is anaphase, they're tearing apart. Prophase, because everything's preparing, sort of starting to condense. Interphase, because everything's just sort of spaghetti at this point. D in this image is telophase, they're tearing apart. And E is metaphase, everything's lined up along the middle. This is not in order, obviously, this is just a random question. This diagram on the right, also not in order, so let's go through each. Diagram A is interphase, nothing has any rhyme or reason. B is metaphase, everything's lined up along the middle. C is anaphase, because everything's tearing apart and going to opposite sides or poles of the cell. D, there is also anaphase, it's just further along in anaphase, we call that late anaphase. E is prophase, it's all kind of preparing, nothing really fancy going on here. It's not like metaphase, which looks similar, but in metaphase everything's lined up in the middle. Prophase, it's not, it's just sort of messy. Here's another anaphase image. Here's another anaphase image, and notice how anaphase can have different portions, and you're looking at a snapshot in time. So this might be late anaphase, and this might be very early anaphase. Telophase, we've made 2 new cells, and this one's telophase as well. You can't really see the label because my face is in the way, but both have formed 2 identical cells. So that's just a little practice activity for you. Okay, so let's talk about cancer. Cancer cells form when you have cells that can't stop doing this process. If mitosis never stops, then you never stop copying and dividing, copying and dividing, copying and dividing. And cancer is exactly that, uncontrollable cell division. So cell division of a certain part of this body, certain cells in the body that just don't stop dividing. That's what a tumor is. A tumor is a lump of cells that literally has not stopped multiplying. So cancer cells are dangerous because these cells never specialize. They never really do what they're supposed to do. They are sort of like the cells that are supposed to be in that area of the body. For example, lung cells or throat cells. But they don't do what they're supposed to do. So there's just a lot of half-broken cells that don't stop dividing, and then you have too many of them, and the body starts to fight them. So that's all we'll talk about for cancer here, but I do want to just hit up some other brief discussions. Cloning is another aspect of just cell division. Now, humans don't clone. Many organisms don't clone, but lots of plants clone. And so asexual reproduction is when an organism is able to produce a total new organism through mitosis. Bacteria do this, carrots do this, bees do this, mushrooms do this, Komodo dragon lizards do this. Identical twins are kind of clones of each other, but the process is very different. And this usually occurs in other organisms when the environmental factors are stable. So an organism will usually clone itself when things are stable. For example, if you have a spider plant and the spider plant has a little baby shoot coming off of it, you'll find that spider plants only do that little budding and have that baby spider plant clone when things are really stable. The temperature is stable, they've been fed and watered, consistently, the plant feels safe, boom, it makes a clone, or in other words, a baby of itself. The advantages of asexual reproduction are that it's quick. You can do it yourself. You don't need a partner, so you don't have to stress about if you like somebody enough or if they like you. You just like yourself, you make more of you. The disadvantages here, I'm kind of cut off here, I apologize, but there's no genetic variation. So the gene pool is really limited. If a disease hits that forest with a bunch of spider plants in it, for example, the whole thing's going to die because all of them are the same. Any mutation that the parent has, all the kids are going to have because you've cloned that individual, right? Okay. So that's it for genetics for now. If you have any questions, email me or comment below. Thanks, guys. Bye.